Hello students, today we are going in detail about construction of a cDNA libraries. For this there is a need to prepare the cDNA, and from this cDNA the genomic DNA library construction is carried out. First we must know what is cDNA. So this is nothing but the cDNA, which is a complementary DNA, and in cDNA library there is a collection, or we can say population of bacterial transformants, or sometimes we can use FAG, from which each mRNA is isolated from an organism, and this tissue is represented as its cDNA insertion into a suitable vector, either plasmid or FAG. But its frequency depends upon the concerned percentage or frequency of mRNA in the tissue or the organism which we are using. Then what is cDNA? This is a copy or complementary DNA, and this is produced by using mRNA, or we can say messenger RNA. Any RNA molecule can be used, but mRNA is of good results. So DNA copy of an RNA molecule is produced by enzyme, and that is reverse transcriptase, or we can say RNA dependent DNA polymerase. This enzyme is obtained from avian mycoblastosis virus, and this is first invented by Temin and Baltimore in 1970. This is the figure that gives you idea about the steps involved in cDNA libraries. So for this construction of a DNA library, we have to use a primer first, and this is a poly T primer, or we can say poly T oligonucleotide. Why this is used? Because the mRNA which we are using, they are having poly A tail. So obviously we know structure of the DNA that A, it pairs with the T. That's why poly A tail of a mRNA that pairs with the poly T primer. Afterwards, this primer which pairs with the 3' end of a mRNA, and from which with the help of a enzyme reverse transcriptase, which is RNA dependent DNA polymerase. So here this poly A tail is added to the 3' end of the RNA to make its analog to the eukaryotic mRNA. Thus afterwards this reverse transcriptase that extends 3' end of a primer. So this is the 3' end of the primer, and it form the complementary strand. And now we are with this DNA RNA hybrid molecule. Afterwards we have to remove this RNA strand. So with the help of RNase H or alkali, mRNA is hydrolyzed, or it is removed. Now we are with a single stranded DNA strand, or this is also called as a single stranded cDNA. But here in this step, for preparation of a double stranded DNA, there is no need of a primer. Why this is so? Because 3' end of this single stranded DNA, it serves as its own primer and provides free 3-OH required for synthesis of its complementary strand, and this is why there is no need of a primer. Then this complementary strand of a cDNA is synthesized either by reverse transcriptase itself or E. coli DNA polymerase, and it generates the hairpin loop in the complementary DNA. Afterwards, this hairpin loop is cleaved by a single strand specific nuclease. So there is a use of again specific nuclease to remove this hairpin loop model, and finally we will get this double stranded DNA, or this is a regular DNA duplex. So in this figure it is clear to you that how there is a need of a primer, and this is with a poly T oligonucleotide because mRNA it is with a poly A tail, and finally we will get the double stranded DNA. These are the steps again for your revision. As eukaryotic mRNA we are using as a template and it is with a poly A tail at their 3' end, and there is a use of a primer like poly T oligonucleotide that they will base pair to the 3' end of a mRNA, and reverse transcriptase extends this 3' end of a primer using mRNA as a molecule, and will get DNA RNA hybrid molecule. In this step we have to remove the RNA strand, and this is carried out with the help of alkaline hydrolysis. Then the 3' end of this complementary DNA serves as its own primer and provide 3' OH. So there is no need to use a primer in this step. And here the formation of a hairpin loop is observed in the DNA. So this is cleaved by a nuclease to get a DNA duplex. Then the another point is a genomic library. This is nothing but the collection of recombinant plasmid. Recombinant means we are having the foreign DNA segment of our interest, and this is nothing but the sum total of DNA inserts that represents entire genome of concerned organism. Here in detail about the steps involved in construction of a genomic library. So obviously first step is to extract the total genomic DNA from an organism. Then we have to cut this DNA into the fragments of the appropriate size, because we have to insert this fragment into the vectors, and this cutting is carried out by the restriction endonucleases. Specifically there is a need of a partial digestion of DNA. Why this is so? Because a partial digestion of a DNA can form the fragments of a homogeneous size, whereas complete digestion is avoided because it creates fragments of heterogeneous size. And for this digestion, preference is for 4 base recognition site is expected, because every 4⁴, that is 256 base pairs in DNA is assumed, that because arrangement of these 4 bases in the DNA molecule is random: adenine, guanine, thymine and cytosine. What will happen if we take the 6 base pair target site? It will observe after 4096 base pairs, so it will be a very large, so use of this 4 base recognition site is expected, and for this preference is for 6 base hexameric target sites. Then the fragments which are produced due to the partial digestion with the enzymes having 4 base recognition site, as they gives you appropriate size for cloning, and that is 23 to 25 kilo base pairs. and the suitable vectors for its insertion are lambda vectors or plasmids, because they are having the insertional size up to this 23 to 25 kilobase pairs. Afterward, the single or mixed digestion with the enzymes like ALU first, HAE third and SAU 3A is used basically for the construction of a genomic library. Same sets of fragments are obtained from a DNA each time when we can use a specific enzyme, as many of the enzymes they are producing the cohesive end, and partial digestion of genomic DNA is subjected afterwards for electrophoresis or sucrose gradient centrifugation. Why this is so? For getting mixture of a fragment of appropriate size, and afterwards this DNA of our interest is inserted into the vector, as I already told you, lambda vectors or the plasmid, because of its insertional size. And by screening techniques we can identify the bacterial colony or the phage colony that contains the desired DNA. So this is all about cDNA. What is cDNA? This is a complementary DNA or copy DNA which is obtained from the messenger RNA. Then the steps involved in construction of this cDNA, and afterwards the genomic DNA library is constructed. In next lecture we will go for the methodologies which we are using for the separation as well as the purification of the DNA. Thank you.